Hi everyone, my name is Pete Regas. I'm a member of the Lambert Lab at Cornell University. And in this session Y05, which is titled Biomaterials and Nanotechnology 2, that's sponsored by the Biological Physics Unit, I'll be discussing binding selectivity of Cas nucleases. To give a presentation outline, I'll be discussing computational approaches for predicting Cas nuclease behavior, from which we will also discuss experimental data gathered from our colleagues for measuring binding selectivity and fitness impacts for several Cas nucleases. And finally, to determine whether there is any broader biological context in which our model can be applied, we'll discuss how we can leverage our efforts for modeling unexpected fitness impacts of dCas binding. And towards gathering experimental data for different catalytically inactive nucleases, we'll first be discussing work that's related to efforts from my colleague David, who is a recent PhD graduate, in addition to the work of Yasu, another graduate student and our principal investigator. And in our first paper published by the Lambert Lab, they looked at CRISPR interference assays in the context of dCas 12A binding, in which the method primarily relied on the thermodynamics of nuclease binding that's informed by statistical mechanics approach, in which a partition function that's statistically weighed in the PAM, reconfiguration rate, and CRR DNA inspection steps that the Cas nucleus undergoes for binding is formulated. And visually, one of the steps that's illustrated in the inspection is given below, in which the Cas nucleus can develop some tolerance to mismatches in the sequence that's inspecting for complementarity. And our guiding question in this presentation is to determine whether the computational model that we will introduce can be applied to other Cas nucleases, which can have energetically different contributions from the PAM, DNA, RNA inspection sequence, and or the reconfiguration rate. And this is very much related to RNA polymerase, which is a molecular counterpart of Cas nucleases, which diffuses upstream and downstream of a DNA target strand when investigating different genomic loci. In the differential equation and first passage time models the following. We're interested in predicting and approximating the first passage time distributions tau of x, and this physically relates to simulations of the protein inspection process that the Cas nucleus undergoes, which is governed by a second order differential equation. The differential equation reads d squared tau by dx squared minus d tau dx, with a prefactor to the first derivative of the passage time distribution term, which is given by this first order spatial derivative of the landscape potential, in addition to a constant -1 over the Boltzmann constant times the temperature term. And we're solving the second order differential equation subject to the initial conditions that tau of n is 0 and tau prime of 0, 0, where n is an integer. And given characteristics of the landscape potential u prime of x that we considered in our March meeting presentation from last year, we know that the analytical solution has the following form. Tau of x is equivalent to the reciprocal of the Boltzmann constant times the temperature times a superposition of three terms, including the absorbing boundary term, the exploration term, and finally an error function double integral, where the functions f sub h sub I that appear as in each of the integrands is a smooth exponential function with a prefactor given by a product of the variance of the height that's given in the landscape, in addition to an inverse proportionality of the height h sub I in the bearer in the temperature T, in addition to an error function. And given this initial value problem that we stated in the previous slide, we can form the following computational workflow. We're interested in obtaining approximate solutions to the second order differential equation, which corresponds to looking at an admissible landscape U, where the admissible landscape U can potentially be a smooth function that exhibits some symmetry in the depth of the energetic well around some particular base pair of the sequence, or there can also exhibit asymmetry in which the Cas nucleus has to interrogate certain base pairs of the sequence that occur later in the inspection process that can be more energetically demanding. And from this initial value problem, we can assemble our first passage solutions to introduce a temperature dependence on our approximations. And in this plot below, we're able to study the relationship between exit time and base pair of the target sequence, in which we plot the base pair versus the exit time. And the ensemble of solution curves that we're able to generate in this plot have the following curvature. And the intercept of the solution of each one of the solution curves with the base pair axis indicates the predicted exit time for the Cas nucleus at the base pair of intersection. In the first passage times exhibit monotonicity with respect to the length of the absorbing boundary. From the smooth exponential that comes into form in the analytical solution that was previously introduced, when we produce our solution approximations in the command line, we can apply our first passage approximation to a wide variety of Cas nucleases. With this methodology, we can also, besides only FN Cas12a, we can look at Cas9, Cas12a, Cas13a, and Cas13b to name a few. Each one of these Cas nucleases has its own characteristic target, tracer RNA, nucleus domain, and PAM sequence that it interacts with. And this allows us to form nucleus-specific characterizations of binding specificity and editing efficiency. And preliminarily, when we applied this first passage model to FN Cas12a, the duration of the passage times over the middle 8 base pairs can be approximated to around 200 milliseconds, which has been experimentally realized as the occupancy time that the Cas nucleus spends in those base pairs when inspecting for complementarity. And we can fashion additional analytical results given the solution that I just raised. The first quantity, or the first relationship that we can form, is that we can look at how to maximize the first passage time given a landscape barrier height and binding temperature, in which we can form our first order integral relation, which is dependent on height temperature prefactors, in addition to one temperature dependent exponential, in addition to an error function like term. In second, we can look at the first passage time fluctuations, which will come into play later in the presentation, in which the first passage time fluctuations take the form of delta ij tau, and this quantity is a function of the binding temperature at position J, the binding temperature at position I, in addition to the height of the landscape barriers at positions I and J, which are respectively given by H sub I and H sub J. And this analytical relationship is composed of absorbing boundary terms, exploration terms, in addition to error function double integral terms, where the quantities, again, that appear F sub H sub I that can be dependent on the temperatures T sub J and T sub I is a smooth exponential of the same form that was given previously. In our workflow for measuring dCas binding fitness changes after binding has occurred is related to the following objective that David examined. We want to determine whether the presence of new genetic material that can be amplified as a virus propagates to genome mutations to the host genome after infection. For the first step of his workflow, he looked at choosing a viral genome of interest, whether it be SARS-CoV-19, influenza, or Zika. And after performing some genome mining, he can enumerate combinatorial target spaces and also measure the dCas nucleus binding activity through next generation sequencing. And we can identify editing target sites for dead Cas nucleases depending on whether the PAM is proximally or distally spaced to the target site. And amongst a wide variety of Cas variants that he included or he studied, he looked at Cas12a, Streptococcus pyogenes for Cas9, and in addition to other variants for Cas9, as well as finally Cas-X. And overall, he was able to obtain hundreds of thousands of testable constructs. To continue his workflow, he also looked at measuring RNA degradation from each combinatorial target, in which several ingredients from synthetic biology come into play. Within this isolated system, circular segments of DNA, we have a Cas13a plasmid system, which consists of a repression mechanism and also a guide target sequence, which helps localize the nucleus to the target editing site. However, counterintuitively, albeit perfect complementarity existing between the target and guide RNA sequences, inspection of the RNA target template confers a fitness penalty, potentially through interference mechanisms related to reverse transcription in the interruption of DNA replication. And for the final step of his workflow, he looked at how to obtain experimental readout and data acquisition, in which he formed a crosstalk map, which is a plot given of the various form. On the x-axis, we have the promoter barcode plotted against the y-axis, which is the CRRNA barcode, in the base 10 logarithms in the number of counts in the condition K in which Cas binding can occur is predominantly localized to the diagonal of this crosstalk map. However, away from the diagonal, the majority of the pairs of the promoters in the promoter barcodes and the CRRNA barcodes are colored blue, which indicates that there's a lower abundance in the log 10 of counts. However, from the Cas nucleus that he tested from his dead nucleus pool, there were other candidates such as dCas9, which exhibit unanticipated fitness effects in which the coloring of the significant abundance of the log 10 of the counts can also be localized away from the diagonal. And we want to form a computational experimental synergy in which we want to determine why there's an expected abundance in the log of the number of counts localized away from the diagonal, which consists of the components I had mentioned before, including RNA polymerase, this dead-Cas nuclease, the cRNA inspection, and the ternary complex that is formed. In our thermodynamic first passage time fluctuations are introduced with the following expectation. We're interested in looking at thermodynamic instability in the first passage times, which can accelerate molecular degradation through singular various components. If we look at the first passage times that are associated with landscape 1 and landscape 2, given the initial value problem that we raised earlier, we can look at another landscape 2, which differs from the landscape 1 above it in the energetic barrier beyond the landscape minimum that's located at base pair 14. And the two primary quantities of thermodynamic interest are the two error integral terms, which appear in the analytical expression for the first passage time fluctuation. In our simplified minimalistic high temperature fluctuations take the following form. From this analytical relation, we can generate either temperature level sets or a fluctuating manifold, and the temperature level sets along the diagonal indicate the equal temperature in which we can encounter thermodynamic disequilibria by going either to the left or the right of this line. Furthermore, the fluctuating manifold is characterized by, in the high temperature fluctuations as a function of the two ambient binding temperatures. And the numerical simulations of the high temperature fluctuations exhibit that the first order fluctuations of the manifold in high temperature manifest in a sharp phase transition, while second lower order fluctuations can be dispersed further along the manifold boundary. And our consequences is that we're able to characterize the landscape-specific fluctuations in addition to characterizing the range of molecular mechanisms that can be damaged with dCas binding, in which if the DNA is destroyed and irreparably damaged, then proteins and other molecular entities cannot interact with this region anymore. And our conclusions is that we provided an analytical view of our first passage time model. We discussed the experiments which contain unanticipated fitness effects in addition to simulations from the analytical solution. And we're interested in building on our further work to further investigate the construct space for more distinguished in characterizations. And I would like to thank my lab for several comments, and please feel free to contact me at my Cornell email address if there are any additional questions. Thank you.